In this topic, we will have two outlines. First one is about the yeast longevity genes research that I want you guys to read on Tuesday's class. And after that, we will look into a new algorithm, which is a combination of hierarchical clustering as well as k-means clustering. So for the yeast, as I discussed with you guys earlier, we are interested in longevity genes. Compared with human, yeast has only about 5,000 to 6,000 genes, so it's much easier. For biologists, they can knock out one by one and then really doing some what we call wet lab experiments and to see if the genes are live longer or live shorter without that gene. But of course, that takes a lot of time and effort to do so. So we want to see if computer science can move into this area and then do some researches or try to make some predictions about longevity genes. And that's what we call a bioinformatics research. So right now, there's only, at the time of the research, actually, only 86 genes were identified. Compared with expected 500 genes, there's still a big gap between those two. So what we have done is this. We try to look into all of those 5,000 genes. And what we're trying to do is that we look into a website that provides the information for what we call a protein-protein interaction. So for example, by looking into one of the genes out of in the yeast, it has totally 463 interactions with all other genes. So there's a lot of interactions. This is an active one with a lot of activities with other genes. What we're trying to be interested is that among all of those interactions, how many of those are actually interact with the known 86 genes that we are interested in. And we will be able to generate a ratio, and we can sort of that ratio to see what other genes has more interactions with longevity genes. In biology's perspective, the more interactions indicating the higher probability that you are also a longevity genes. So that is our assumption. And because of that, we're actually using what we call a z-score concept to filter out only 5% of the genes that are highly interested in. And therefore, we pick up 197 genes. And what we're going to do next is that we put this 197 genes together with the 86 non-longevity genes together. And then based on their microarray data set, we apply hierarchical clustering. So what is that kind of data set? The data set is something looks like this. Looks familiar to you guys? This is actually the file I want you guys to work on exam number 1 or the midterm exam. So actually, each gene on the left side, these are the gene names. And then for each row, each column actually, is the experiment results of the genes. So based on the experiment, what is the response for that experiment? And we can have a number to record it. Because they are different experiments, the values are actually very different from each other. That's why we need to do the normalization, just like you guys did in the midterm. Once we have normalized those, they become 3 values associated with each genes, and based on this, for all of those 283 genes, we do a hierarchical clustering and then try to find the clusters among those. And therefore, what's happening is this. Remember, for hierarchical clustering, based on the operations that we do for every iteration, starting from all of the points that are considered as 1 point, we try to find 2 points to merge together. Only in this research, 2 or more points are considered as a cluster. So you can see that based on different number of iterations, for 10 iterations, we have 8 clusters. For 20 iterations, we have 12 clusters. For 30, we have 18. For 40, we also have 18 clusters. So we have this figure to keep track of how many clusters are being generated. And we are actually, in this presentation, I'm going to show you the interesting things on the number of iteration of 40. So basically, when we have 40, we actually end up with 18 clusters. So while we try to circle out or presenting all of these clusters, there are 5 of those are not shown over here. We try to cluster all of the genes together, and then I want you guys to take a look of this one. So saying that for the clusters of, let me make it bigger. For the cluster of cluster number 1, there are 1, 2, 3, 4, 5, 6, 7. 7 genes are put into the cluster. The one with the star sign indicating they are the non-longevity genes. So basically, by looking into this one, you see that these clusters has these amount of genes, and 3 of them are non-longevity genes. Compare with cluster number 3. This is actually very interesting. In this cluster, almost every single one is a non-longevity genes except this one. And obviously, this suggests that this gene is actually very similar to all of this. And that's why we can predict that one as a non-longevity genes. I will continue my slides in the next video.